GI tract. It is actually a very long topic. So, and the presentation is also long. So, I will focus on the more important points. But for study purpose, you should know about these organisms as well. Some of these organisms must have been covered in first year in parasitology section. So, today we will discuss about the GI helminthic infections. We will discuss about the common helminths that cause infections in GI tract, compare their characteristics, important characteristics only, which are important for their identification and diagnostic purpose. And we will discuss how we diagnose them, and in some cases we will discuss about treatment also. Treatment is actually the easiest part for most of the helminthic infections. Diagnosis is the most difficult part. Before I proceed in this topic in detail, these organisms are unique in the context that the normal antibiotics, for example, if you take any of these helminthic infections, they will not be treated by any antibiotic. We need a specific drug for them. The more common drugs like albendazole, mebendazole, praziquantel. So with simple, in most of the cases with single dose, you can treat the condition. But many times diagnosis is missed because these infections are not very common. So we will focus on their clinical features also in some cases, which are important to differentiate them. If you see, they are actually included in under parasites. In parasites, we divide them into two major types, protozoa and metazoa. In protozoa, we have unicellular organisms like, for example, Entamoeba histolytica, some of the flagellates. In multicellular organisms, we have arthropod and helminths. So, helminths come here. They are multicellular parasitic organisms. They are broadly divided into two types, nematohelminths and platyhelminths. Nematohelminths are roundworms, platyhelminths are flatworms. These flatworms or platyhelminths are further divided into two types, cystodes and trematodes. These roundworms, they are commonly known as nematodes. So we basically have three types of helminths: nematodes, cystodes and trematodes. And then further they are divided into different class species. This is the class, and after that there is genus, species. Now, as I will go into the details of these organisms, so you will know it better in later slides also. But in general, nematodes or roundworms, if you see, they appear round in cross section. They have body cavity and straight alimentary canal and in anus. We will discuss these features also. Common examples are Ascaris, Ankylostoma, Necator americanus, Strongyloides, etc. They are also known by their common names. We will discuss these common names individually when we will discuss the organisms. Platyhelminths or flatworms, they are divided into two types, cystodes and trematodes. In there they are dorsoventrally flattened. They are flatworms. So this is the major difference between nematodes and flatworms or platyhelminths. There is no body cavity and alimentary canal. Even if it is present in some cases, it is not actually complete. Anus is absent. So and if we further divide them, what is the difference between these tapeworms and flukes or cystodes or trematodes? If you see the red part, they have got segmented body, and trematodes have got non-segmented body. Beside this, they have head with sucking organs. They are leaf shaped. Sometimes they are, the name leaf like flukes is also used for them, with two suckers but no distinct head. They have an alimentary canal and are hermaphrodite. Hermaphrodite, what do you mean by this term? They have got both, this is true for cystodes as well as trematodes. They have got both functions of males and female in same organism. Example are Taenia, tapeworm, Echinococcus, etc. Flukes we will discuss in detail along with all these organisms. They include blood flukes, liver fluke, intestinal fluke, etc. So all of this will same things are written over here. The general feature of helminths. This is true for all three: nematodes, trematodes, and cystodes. They are multicellular organisms, eukaryotic. Macroscopic, they can be visualized with naked eyes. We don't need microscope for their visualization. Most of them have got these stages: egg, larva, and adult complex life cycles. It depends on individual organism actually. Infective stage is mostly egg or larva in all the helminths. Definitive host is defined as the host which harbors the adult stage of this organism. For any organism which causes infection, definitive host is one in which sexual phase of the cycle takes place, life cycle. So definitive host is that harbors adult stage. Intermediate hosts may be there, may not be there, maybe more than one, maybe more than two. It depends on individual organism. Modes of entry, mostly ingestion. In some cases, penetration of skin or mucous membrane. How they cause damage? They can cause this formation of granulomas inside the organs, as we will discuss in some organisms. They can block the intestine, they can block the bile duct depending on which type of organism. Sometimes pressure symptoms can happen due to the formation of granulomas. Besides this, necrosis, etc. can happen. So multiple things can happen. Indirect damage, they can cause inflammation, hypersensitivity, and mucosal changes. One thing which is not written in this whole presentation is the presence of pruritus or itching due to this hypersensitivity. And in most cases, the patient may come with complaint of pruritus or itching. As I will discuss these organisms in detail also, this is a tabulated form of some of the helminthic infections in summary, like tapeworms, hydrated disease, blood fluid, liver fluid. I will come back to this maybe later when individually I have discussed these. Nematodes. Now coming to first. Nematodes, commonly known as roundworms. Their body is elongated, cylindrical, and unsegmented. Like you can say a wire or cable. I will show you one picture, real picture. So see this. This is a typical roundworm. Many roundworms are there in the hand. So they are cylindrical, unsegmented, elongated, bilateral symmetrically. Sexes are separate. It is different from other types, cystodes and trematodes, and male is smaller than female. Alimentary system is complete. This is also a difference from the trematodes and cystodes. Size is variable, less than 1 centimeter to about 100 centimeter. Important organisms: Ascaris lumbricoids, Ankylostoma duodenale, Necator americanus, Strongyloides, Stercorylus, Trichinella spiralis, etc. They are given common names also. For example, roundworm, most of the time we use term for Ascaris lumbricoids. Hookworm, there are two types or two organisms, Ankylostoma duodenale and Necator americanus. Sometimes we use the term porkworm for Trichinella spiralis, and we use the term pinworm for Enterobius vermicularis and whipworm for Trichuris tricura. This is the general life cycle of this intestinal nematode. If you see eggs of the nematode, they get ingested, and in some cases, for example Trichuris and Enterobius, they remain in the intestine and develop there as adult worm, and the cycle is repeated. In some cases, for example Ascaris, these larva formation is there and they enter bloodstream, and through the bloodstream they reach lung and they pass through the lung, and again they come back into the intestine. This will be discussed in detail also.
So the sign here is the definitive host.
What?
The definitive host.
Yes, what?
Sign.
No, definitive host mean organism in which it is having adult stage. It can be human. We will discuss individually when we will discuss organism. We will discuss about which one is definitive host. But in general, you can remember definitive host is one in which this organism goes sexual cycle, or there is sexual development or adult stage. In some of the cases, eggs, larvae hatch from the eggs, but they don't enter the blood stream. They are outside the body, for example in water bodies, and from there they develop into different types of larvae like filariform larvae, and they penetrate the human skin, and through the skin they enter into the bloodstream. These things will be clear as we will discuss individual organism, because this only general life cycle. Now coming to individual Ascaris lumbricoids. Yes.
So the last side you said it will penetrate the skin.
Some of the organisms.
From where? From outside to inside, or the opposite?
Outside.
To inside.
Yes, it is in the water or sand or somewhere. When somebody is walking barefoot, they can enter through the skin. This will be clear as I will tell you in which organisms this thing happen. It is different for different organisms. That was only general life cycle. So coming to Ascaris lumbricoids, it is more common in rural areas. I will focus on the important point. As you can see, this is the picture. These are many Ascaris actually in the hand. After deworming or some procedure, they have come out of the intestine. They are more common in tropics and subtropical area, more common in children than adults. They are found in jejunum and upper part of ileum. This Ascaris is, we use the term for disease as Ascariasis. It is considered as most common intestinal parasite. It can be ingested from vegetables or waters which are contaminated by eggs. Now what happens in the life cycle? This egg goes inside the body. Now if you see in all these slides, there are two things. One is I is infective stage, D is diagnostic stage. So remember this I and D, because these are the two important stages. When it goes inside, as you can see here, this is 7th part. It actually goes from intestine, it penetrates the intestine and it enters the blood circulation, and through the blood circulation basically it enters the lung, and there it again causes sign symptoms related with, sometimes related with we use the term pneumonitis for that. There is inflammation in the lung, but basically this adult worm is formed and remains inside the intestine, and this female, it actually through the feces it lays egg in the stool, and this stool, from a stool these eggs come outside and they can again contaminate this water or food or anything. So this cycle goes on like this. So coming back again to this part, adult worms grow and reproduce in the small intestine, do not attach to wall and derive their substance from ingested food, whatever the person is eating. Many times these organisms will remain inside the person, but the person will not know it. He remains asymptomatic. An adult female can produce up to 240,000 eggs per day, which are passed in feces. Lifespan of the adult is about 1 year. They are cylindrical in shape, as you can see here. Male is smaller and distinctly more cylinder than female. So which one is male, which one is female in this? Yes. On the tip of the head there are 3 lips. These 3 lips arranged as a Chinese word like this. They have a complete digestive tract, and reproductive organs are also there, which are tubular. These are the 2 main types of eggs which can be found in the stool sample, and we use these eggs for diagnosis of Ascaris infection: fertilized egg and unfertilized egg. So there is a distinct difference in these two, if you can see. If you see, eggshell is thinner than the fertilized, it is thicker. Here the layer is thick. Here even the layer is not completely visible, it is very thin. It contains highly refractive granules. It cannot be made out in this picture very clearly, but if you see, this is the difference. As you can see, this ovum is very clear in this. There is a space also here which is clear in fertilized egg surrounding ovum, and we have thick layer. In this, no such ovum you can visualize, and even the outer layer is thinner. So that is the difference, main difference between unfertilized egg and fertilized egg. Why I am showing this picture? Because this is actually what we visualize for diagnosis. So yes, as you know that this organism passes through the lungs, so it can lead to symptoms of pneumonitis and sometimes asthma attacks. And we have one more thing, Loffler syndrome, which is not exclusively associated with this, but some other parasites also, in which we see eosinophilia along with symptoms of pneumonia, cough and allergy like urticaria. So Loffler syndrome we can see in this. If you got any patient with this complaint of itching and pruritus along with the then you should actually do this test for eosinophils, which we do as absolute eosinophil count. Even if you are not thinking in this term, that if you are thinking that this may be allergy, but allergy is coming back again and again, and there are some abdominal upset or something like that, these symptoms are there, then we should think in this term also, that this can be Helminthic infection, because it is very common actually. Eosinophil count increases in many Helminthic infections, including Ascaris infection. This is a larvae which can be seen only on biopsy, as you can see here, larvae in lung. It cannot be seen in stool, they form here. Adult, the presence of few worms may be asymptomatic. Most common symptom is vague abdominal pain. Large number of worms may lead to malnutrition and or obstruction. Migration of adult worms may cause signs and symptoms of perforation, peritonitis when these worms move in the body. They can also aggravate this appendicitis and may lead to actually appendicitis, but there is no definitive cause of appendicitis as such. No intermediate and reservoir hosts, only they are found in which organism, which species? Human beings. So only transmission is from human to human. If you see this cycle again, so these eggs, they go into the environment and they again come back to the human being. There is no other host. There are 2 phases in Ascariasis: blood-lung migration phase of the larvae and intestinal phase of the adults. Microscopic examination of eggs in the stool, you know two types of egg can be found, unfertilized and fertilized. Picture I have shown you. Sometimes larva can be seen in sputum, but it is rare. Now treatment is very simple, albendazole or mebendazole, single dose for albendazole, mebendazole 100 milligram orally twice daily for 3 days. Prevention, all the general preventions for any disease associated with fecal-oral route. Now coming to hookworm. Hookworms, there are two major types, this Ankylostoma duodenale and Necator americanus. The most important thing to remember in hookworm is anemia. Anemia, it leads to anemia. So clinically, the patient may present with anemia and iron loss. They are basically blood sucking worms. They go, they remain in the intestine and they suck the blood from intestine. So adult worms have expanded mouth with hard hooks and triangular cutting plates, and anticoagulant glands help in attachment to intestinal mucosa. If you see, this is the mouth of Ankylostoma duodenale. 4 teeth can be visualized, 4 cutting teeth on each side, and this is Necator americanus. These are the cutting plates. So this is the bigger picture of the mouth. So what happens when they are inside the intestine, they keep on sucking the blood and there is blood loss obviously, and this leads to anemia. Larval penetration is associated with local dermatitis, ground itch. How they actually penetrate through the skin. Life cycle of Ankylostoma duodenale and Necator americanus. If you see, this is the beginning here. Basically eggs, they are this eggs may develop into 2 types of larvae, rhabditiform larvae and filariform larvae. This filariform larvae is the infective form. When a person walks barefoot on moist soil, for example beach or near the pond or river, then these larvae penetrate the skin, and then through this skin they enter into the blood stream, and they basically goes into the intestine later on, where they develop into adult worms. This whole is explained in here also, I will explain it. So this is what actually local dermatitis or ground itch. The point from where they enter, the person may feel there is something, but most of the time he will not know it that this organism has entered into skin. Now this is the picture of eggs of Ankylostoma duodenale and Necator americanus. They, as you can see, these are simple. Actually, there are 4 inside this, 4 parts or circular structures. It is not visualized actually in this, and this is not a line diagram. So actually how they look like, it is very simple to identify them. And if you see, the size is also important for all these organisms, as when we see the egg in stool, we compare the size. So it is comparatively bigger than many other cysts or eggs. And this is the picture. Important part is this, I have told you already, the teeth structure, and in this case cutting plate through which it gets attached. This same things are written over here about life cycle. Humans are infected when filariform larvae in moist cell penetrate the skin, which can be through feet or leg, and they reach the blood and then to the lungs, migrate into the alveoli and up to the bronchi and trachea, and then are swallowed back into the intestine. So this whole thing, the same thing happens for ascaris also, that when it enters the blood goes to the lung and then alveoli, and this is common for all these, and then trachea, and then they come back into the intestine through the, when they are swallowed through the esophagus. They develop into, yes.
When they reach the lungs and go up to the trachea and be swallowed, what is the size of this?
This actually, they are not in, they achieve this adult stage only when they reach intestine. They are basically in the form of larvae at till this stage. And when they reach intestine, they will develop into adults because the size is very big. If you see, you will understand from this, the size of X is only in micrometers, okay, but the actual organism size is, for example, in this case 1 centimeter, but in ascaris it is even bigger. So it is not like that the organism is moving, only these larvae and eggs are in micrometer size, they are going inside and they are developing into adult forms.
So there is two phases at the beginning. You said one of them is the affecting one.
In which one?
The right side. Two different.
Yes.
These two.
Basically, filariform larvae, there are two, rhabditiform larvae and filariform larvae, two stages of larvae. So this filariform larvae is the infective stage. This will be, if you see here, all the things are written. They develop into adults in the small intestine attaching to the wall. So what is important for hookworm, I have told you already, they feed on blood in the intestine from the capillaries of the intestinal villi. Again, eggs are passed in the feces. Eggs first develop into non-infectious type feeding rhabditiform larvae, and then into third stage infectious non-feeding filariform larvae. Actually, I am not going into detail of life cycle because it is not possible to cover all the organisms. So just I am focusing on important stages. So you can remember for now that there is one rhabditiform larvae stage and one filariform larvae. So infection happens due to this filariform larvae stage, which can penetrate the skin to complete the cycle. Diagnosis is very easy, but actually difficult also if you don't suspect the organism. If you order for stool examination, then you can see the hookworm eggs. In any patient of anemia with some symptoms of helminthic infections, for example, abdominal discomfort and all these things, you can order this stool examination also, because many times this hookworm infection can be the cause. You should measure hemoglobin if the patient is presenting with anemia, serum ferritin, iron, etc., and exclude other causes of anemia. All the things for prevention and control are general things. As you can see, this can penetrate through the skin, so a person should wear this footwear, especially in damp soil. Children have the habit of soil eating, so they should be discouraged. In some communities where this is common, the drugs can be given. Now coming to Strongyloides, stercoralis, here also the infection happens through skin penetration. And if you see, it is smallest pathogenic nematode with only 2.5 millimeter size. Its shape is something, it looks like a snake in picture. So again, symptoms and signs. As you can see, anemia, constipation, cough, diarrhea, eosinophilic pneumonitis. So these sign symptoms are mostly common for all the infections and not exclusive to Strongyloides stercoralis. This is the stage which we can visualize on this stool examination. Rhabditiform larvae can be seen in stool examination or duodenal aspirate. And this is the life cycle for Strongyloides stercoralis. Here also, if you see, all the things are same, penetrate the skin, reaches bloodstream, lung, then comes to trachea, pharynx, it is swallowed back, then acts in the mucus, they goes into the intestine, and where they develop into adult male and females, and again this female lays the egg which can go into the environment, and again the cycle of infection goes on. So for all of them the cycle is almost same, treatment is also same, albendazole, mebendazole. Now coming to pinworm, Antrobius vermicularis. It is important from one aspect. I will tell you what is that aspect. Commonly known as pinworm, threadworm or seatworm, humans are the only host for Antrobius, common in kindergarten and primary school students. No intermediate host and reservoir host. Male die after mating female, and those are rarely seen. It can be seen with naked eyes also. This is the picture of this pinworm that we see, eggs of pinworm. If you see, it has got a typical plano-convex shape, and it looks like a lens, plano-convex lens actually, and typical D-shaped. And the most important thing in this is regarding its it's a part of its life cycle, is anal pruritus. What happens, the female this Antrobius, it goes towards the anus or outside the intestine, especially in the night time, and lays egg in the anal region of body. So we get perianal pruritus, perianal pruritus or pruritus around the anus region. Nocturnal migration of the gravid females from the anus and deposition of eggs in the perianal folds of the skin. Patient may feel restlessness, nervousness and irritability. So this is the very common thing, and it is different from other organisms also regarding its diagnosis. So how do we diagnose it? As you know, I have told you that these eggs are around the anus. So we can take the sample from anus region. How we take the sample? We can take through anal swab, or we use a cellophane tape. We just simply put the cellophane tape over this like this, as you can see here, and if there are any Antrobius, pinworms, eggs, then they will come into the tape, and then we can, with the help of microscope, we can visualize them.
Nothing with stool comes out.
What?
No eggs come with stool.
Eggs can come with stool also, but this is the recommended method. You don't need stool sample in this. So cellophane tape method is important for diagnostic purpose in this. So in this organism, remember two things which are very important. What is the name of pinworm? Antrobius vermicularis. What is the main feature, clinical feature? Anal pruritus. How do we diagnose it? Through cellophane tape method. Yes.
What is this?
Cellophane tape method means we actually take the sample around the anus, from especially in children, and when you put the cellophane over that place, then the organism, that is eggs of organism actually in this case, they will come into the tape, and you can visualize it with the help of microscope. And if you see, one very important thing, they can be seen by naked eyes also sometimes. So you can get the worm also. So it is possible, but most of the time we see only eggs. Albendazole, mebendazole for whole family. This is true for others also, that when you get helminthic infections, then it is better that you give treatment to other people also in the family, because it is possible that this infection is there in whole family, especially in this case pinworm. Now coming to Trichuris trichiura, it is also known as whipworm. Again here, as you can see, it lives in large intestine, may lead to some complications when there is heavy parasites, that is large number of parasites are there, may lead to diarrhea, rectal prolapse and anemia. It has got this typical egg appearance. As you can see, there are two sides at the end, elliptical barrel shaped with mucoid plugs. So these are basically mucoid plugs, and this typical shape you can remember for Trichuris trichura, and the adult looks like this, whipworm.
What's the lifespan of the worm? How many days?
Which one?
Trichuris.
Trichuris, for all of them the lifespan is not much actually. It depends on individual organism. If it is not mentioned, then maybe it is not important in this. Maybe few months or weeks, not more than that. But what happens, important thing in this is to understand, if you see here, it can survive in external environment for 2 to 3 weeks. But when it is adult, that is it becomes adult in the intestine for all these organisms, they can remain inside the intestine for very long time. If you don't give treatment, they can stay there, especially for example roundworm, Ascaris. Many people will not know it, but they are harboring it for maybe 1 year or 2 years. So this is possible. Whipworm, Trichuris trichura, I will not go into this. Important thing for Trichuris trichura, there is nothing much in this, so I will leave it. Now coming to Trichinella spiralis or Trichinella spiralis. This is unique in some aspects, that it is found in mussels. It can be found in mussels. So it is also known as sometimes porkworm disease, trichinosis, trichinosis, etc. It is zoonotic disease. Why it is zoonotic? Anybody can tell what is zoonotic disease?
Best for animals. Best for animals.
Yes. So it is passed between human and animals. The habitat, females in mucosa of small intestine. The same animal acts as final and intermediate host. So if you see, the picture is not very clear, maybe due to light, but it is thread like. And important aspect in this, that is most important regarding this larvae is encysted in skeletal muscle cells. So this is unique in this regard, that its larvae is found in the striated muscle cells. So for diagnosis, again, if its larvae is found in muscles, then stool sample will be of less use. If you can do the muscle biopsy, you can see the larvae in muscles. If the patient is having diarrhea, then adult and larvae may be found in feces. For diagnostic purpose, ELISA is important. Treatment is same, albendazole and mebendazole. Yes. So if you see, this slide is very important in some aspects. Some of the nematodes which can infect via skin penetration, you know there are 3 now: Ankylostoma duodenale, Necator americanis and Strongyloides stercoralis. Orally infected, Ascaris, Antrobius, Trichuris, Trichinella. Lung is important in these, like that you can remember. This is the comparative picture, as I have told you in that sometimes we see the size also. Size is important for us because we compare the size for adult worms also and also for eggs. These are the worms. As you can see, Trichinella are very small, and this is Trichuris, and it is not written over here. Maybe this is the biggest one is roundworm. This is the summary, some of the important points about nematodes. So from this slide you can get many important points. If you see all the organisms I have discussed, their common names: Trichuris, whipworm, Ascaris. There is no common name. This is the name of disease. Antrobius, pinworm, hookworm, ankylostoma, and Necator. So if you see, mode of transmission is mostly ingestion of eggs. In two of the cases, hookworm and Strongyloides, this is penetration of skin. So this is different from these three. In Trichinella, it is due to undercooked meat. This is the, and how we make the diagnosis. This is unique for pinworm. I have told you, you can see the eggs on skin, perianal region, you can get the eggs. For others, mostly it is eggs in stool. In Strongyloides, larvae in stool. In Trichinella, as it infects the muscle. So, yes, large encysted larvae encysted in muscles. Serology, also sometimes we can see this eggs in stool. Important stage associated with human beings. In Antrobius, I have told you this thing, that female worm goes out at night and lays the egg around the anal region, causing itching in the region, perianal itching. Important stage outside of human, there is none. Trichuris, if you see, worms in colon may cause rectal prolapse. Leave this. Eggs survive in environment. Ascaris, eggs survive in environment, there is no other stage. Ankylostoma, again, the larvae forms outside and filariform larvae penetrates from there. Strongyloides, again the same thing. Trichinella may be found, this larvae is found in muscle of pig. So because I have not gone into detail of this, basically what happens, people who eat pork, they ingest this larvae in their food, and this larvae goes and forms penetrates, and in the whole life cycle it goes and forms in the remain inside the muscle. It is somewhat similar to Echinococcus, which I will discuss. Now coming to, you have class at 9 o'clock? I will go quickly. Cystodes, these are the general features of cystode. They are all of cystodes and trematodes, both are flat organisms. Major difference is they are segmented, trematodes are unsegmented. They are tape-like, trematodes are leaf-like. So I will explain the differences here itself. They are hermaphrodites. Mature segment contains both male and female sex organs. They have suckers in their head. Alimentary canal and body cavity are absent. Excretory and nervous system are not developed. This can be divided into four major parts: scolex, neck, proglottids, and strobilia. These are the suckers, hooks through which it gets attached, neck, and in this picture you can see strobilia, and this hole is basically scolex. When you see it in context, how it, in general how it looks like a tape. If you see, it's thin tape. So that is why the name tapeworm. Scolex has a head with attachment size, suckers, hooklets through which it gets attached. Neck is the portion just after the head. Then we have in this organism proglottids, body segments with testes and ovaries. Strobilia, the chain of proglottids constituting the bulk of the body of adult tapeworm. So in general, you can remember these things. Worm grows by adding new proglottids from its germinal center next to the scolex. The oldest proglottids are at the last end, distal end, are gravid and produce many eggs, which are excreted in the feces and transmitted to various intermediate hosts such as cattle and fish. Humans usually acquire the infection when undercooked flesh containing the larvae is ingested. So, and in this we have four important organisms. Out of this, Taenia solium, Tinea saginata and Echinococcus granulosus are more important. And we have Typhlobathrium latum also. They are associated with specific animals. So I will explain them together. The most important thing in this, if you see the name Tinea solium and Tinea saginata. Tinea saginata is sometimes also known as beef tapeworm. Tinea solium is also known as pork tapeworm. If you see, this is the life cycle of both Tinea solium and Tinea saginata. Here, if you see, for example, this is adult tapeworm, and the eggs are coming out from this. Humans, yes, we are in this part. This is adult tapeworm. For example, the eggs are coming out from this. This basically remains here in this case it is in human being. So when these eggs, these animals, they are eating something, grass or any food from external environment, these eggs will go inside these organisms. Okay, they will eat along with their food. Then after that, what happens? These pigs and cattle are infected when they ingest either the eggs or proglottids in human stool. You know, pig can eat the stool also. They can get from grass or water which is infected with human stool. So there the organism, this organism goes and develop inside these animals, and basically it gets there is cyst formation in their muscles or body parts. In case of Tinea solium in pig, in case of this cow or any beef in Tinea saginata. So when a person who is eating this, they will eat the muscles, and again this will come inside the body, and it will here this there are many, actually there is a whole long cycle. There will be excistation, and it will develop ultimately into adult worms, and it will get attached to the intestine and may remain there for long time again. So important thing in this, that a person is eating the animal flesh, and in the intestine the organism is forming. Now the other important thing, more dangerous disease. This is also a disease, tapeworm infection. That happens only in case of Tinea solium. A person who is infected with this egg himself directly. So what happens? This life cycle of animal happens in human being. That is, cysts are formed in the body of human being, not in animal, because in this cycle actually animal is not involved. Here the eggs from tapeworm are directly going inside the body of human being. So this is a more dangerous condition, and this is known as cysticercosis. Cysticercosis, and it can infect the brain, eyes and other body parts also. When it infects the brain, it is given the name neurocysticercosis. So cysticercosis condition, it happens due to ingestion of eggs. The same thing is here written over here. Note, however, that there are between the humans are infected when they ingest the eggs of Tinea solium in food contaminated with human feces, and the eggs differentiate into cysticerci primarily in brain, eyes and skin. So we have the two conditions associated with this Tinea solium, and again the same, as there is very less time, so I cannot go into the details now. Adults attach to the intestinal epithelium and they develop there. Now for if you see Tinea saginata and Tinea solium, important thing is infective stage. For Tinea saginata, Cysticercus bovis, Cysticercus cellulosi. Cysticercus bovis means the cyst which is formed in the muscle, for example, of animal and like cow, and Tinea solium, Cysticercus cellulosi, cyst of the in pig muscle. And when it is coming from human to human, then the infective stage is egg, when the human ingests the egg. In the two cases, the disease is teniasis, in this case cysticercosis, which is a more dangerous condition. These are the hooklets, rostrilum, suckers. Through the hooklets it gets attached with suckers also. This is the Tinea proglottids, egg. This picture of egg is very important because this is known as hexacanth embryo. 6 lines you can see, 6 hooklets, they are basically hexacanth embryo. This type of egg you will find in Tinea solium, Tinea saginata and Echinococcus granulosus also. This is common for 3. Teniasis may lead to allergic reactions also, vomiting, diarrhea, obstruction of the intestine. Cysticercosis may lead to formation of cyst in the brain and eyes. Diagnosis: teniasis, anal swab to find eggs at perianal region, fecal examination to find segment. In cysticercosis, as the infection or cyst formation is inside the body, for example brain, then you need to do X-ray, CT, MRI, etc. and other relevant investigation depending on the place of infection. Echinococcus. Echinococcus, if you see, is also known as dog tapeworm. In this case, human beings are accidental hosts. Echinococcus granulosus causes unilocular hydrated cyst disease. There is one more organism which is not important because rarely we see a disease due to this, Echinococcus multilocularis, which causes multilocular hydrated disease. Echinococcus granulosus is composed of calyx and only 3 proglottids, making it one of the smallest tapeworms. Dogs are the most important definitive host. So it mostly is seen in dogs. It remains in the intestine of dogs. The intermediate hosts are usually sheep. Humans are almost always dead end intermediate hosts. Accidentally they acquire the infection by consuming food or water contaminated from dog feces. So if you see, the life cycle goes for sheep and adult like this. But sometimes when this egg or this hydrated cyst, it comes in the human body, as it goes into the sheep, then there is cyst formation in the human body. If you see, dogs are the definitive host and contain the adult tapeworm in the intestine. Sheep are important intermediate host and ingest the eggs in dog feces. Hydrated cyst containing larvae form in the sheep. Humans are accidental intermediate hosts. So if sheep is not eating and this this egg comes inside the human body because of this dog feces contaminating water or food, then there is cyst formation in the human body. Cysts are formed in different organs. Most common organ which is affected by this hydrated cyst formation is liver. In liver, hydrated cyst is very common. Besides that, it can form cysts in lung, brain and bone also. This is a picture showing hydrated cyst in the brain. It can resemble in clinical condition with any carcinoma or malignancy of this head. So it is very important to differentiate it. This is the summary of the mode of transmission, main sites, diagnosis, treatment. As you can see, this The treatment is almost same for all. Praziquantel is more common except for Echinococcus granulosus. In this case, sometimes we need surgical intervention also. And also other anti-helminthic drugs, albendazole can be used in many cases. Now coming to flukes. Flukes, they are of many types: blood flukes, liver flukes, intestinal flukes, lung flukes. And they are unsegmented. I have told you leaf-like structure already. Alimentary canal is present but is not complete. Absent, anus is absent in this. So something about this. Schistosoma. Schistosoma also known as blood flukes. They are of different. They are different species: Schistosoma mansoni, Hematobium japonicum, depending on the geographical area. Schistosoma hematobium, it is mainly associated with urinary schistosomiasis. We will not discuss about this. Intestinal schistosomiasis like Schistosoma mansoni and Schistosoma japonicum. It can lead to this splenomegaly and hepatomegaly also. And there can be blood in the stool due to infection of Schistosoma japonicum and mansoni. Here, just I will tell you the diagnostic stage. We can see these eggs here, and the infective stage is Cercaria, which again penetrates through the skin. The clinical condition is mainly due to inflammatory response. You can also see diarrhea, abdominal pain. There is liver is affected and spleen is also affected. You can see a case of hepatomegaly or splenomegaly due to this schistosomiasis, but not very common. Now, if you see the pictures, Schistosoma hematobium are oval with and they have a spike at the tip. Mansoni has a spike on the side. So this is how we differentiate these schistosoma. Praziquantel is effective against all species. Prevention and control is same. Fasciola hepatica, liver flukes may affect this. They can actually affect liver. Again, if you see all of them, their shape is like leaf and they are flat. Humans are accidental definitive host. Again, the infective stage is metacercaria. May lead to blockage of this bile ducts, and as they can reside in the bile ducts, and there can be condition due to this bile duct obstruction. Adult has a flat leaf-like body. They have got oral and ventral suckers. So as you can see, metacercaria sometimes can go inside the body. This is the infective stage, metacercaria, and diagnostic stage is we see the unembryonated egg. You have class with whom? Which class? Subject? Now? Surgery? Okay. Severe infection. So this is actually not much important, but you can see the diagnosis is same. X in stool sample. Serological test can be done. Ultrasound, we can see the fluke itself through the ultrasound. Intestinal fluke found in this intestine, name is Fasciolopsis buscae. It can also come through the same uncooked water plants or through water directly, and metacercaria is the infective stage. This is the egg which we can visualize and is useful for diagnosis. And treatment is praziquantel. This is the general things we do for helminthic infections. For stool examination, macroscopic, microscopic, and other techniques. So we basically make smears. These are the important things: direct saline smear, iodine smear, or we do the concentration techniques using different methods of sedimentation and flotation. So in this, remember, for this direct examination, iodine smear is important, saline smear is important, and concentration techniques, two major types, flotation techniques and sedimentation technique. So with the help of iodine smear, we can actually see cyst morphology in details, and that is why this iodine smear is important. Sedimentation, we use two main methods, saline sedimentation and formol ether method. And in this sedimentation, as you can see, some of the eggs, they will sediment at the bottom. And these are basically very simple procedure. We use saline and stool sample, emulsify it, and just pass through this gauze, and we get the sediment. In this formal ether method, we used ether along with this, and sediment we see here, and formalin we also use. Flotation concentration, we use this in flotation concentration technique, different things, and with the help of this, the eggs float on the surface, and we can say the different methods are saturated saline method, zinc sulfate method, Sheether's sugar method, etc. No need to go into detail for all this. That's all. So whatever I have discussed in detail, organisms or their life cycle or points about the life cycle, so these are the more important points. If I have left it in the presentation, then that is not so important, okay? But it is better if you read all the things from textbook also. Which book you are following? Any book you are following? Lipin Quartz? Or ridiculously easy? Okay, just follow any book, any textbook. Okay, thank you.